Hi there, I'm John Lovell, coming to you from the Hudson Alpha Institute for Biotechnology in Huntsville, Alabama, and the Department of Energy Joint Genome Institute in Berkeley, California. And today I'll be talking about our efforts to build plant genomes and analyze multiple genomes within specific species to better inform breeding decisions and understand traits of interest. As many of you know, the state-of-the-art in plant genomics has recently transitioned from a single reference genome representing the diversity of an entire species to more accurate multiple reference genomes that either span the diversity within a single individual, like an outbred assembly, or across the diversity with many genomes represented. While this is exciting and obviously represents a significant improvement in our understanding of the genetic diversity of species at the genome-wide scale, it has proven to be a challenge to integrate the multiple reference genomes into existing population and quantitative genetics designs. So a toy example of this comes from our work that started about 8 years ago in Panicum hallii. Panicum hallii is an extremely drought-tolerant relative of switchgrass and lives in the American desert Southwest. We took two genotypes, the HAL2 genotype, which is extremely drought-tolerant, and the FIL2 genotype, which is less drought-tolerant but grows much faster, and built whole genome sequences and annotations for these. We then crossed these, produced an F1 that then was selfed to produce an F2 population, which we could use for genetic mapping. Shown below is a profile of the number of genes that are affected by trans-regulatory elements across the genome. You'll notice that there are several hotspots where a single locus affects the expression of hundreds of genes downstream. In particular, that one on the left of chromosome 3, labeled 3A, regulates the drought response of around 250 genes. We were really interested in what the causal variant was. We had initially mapped reads from one reference genome to the other and generated a SNP database. But this didn't really get at what we were looking for, which is a full understanding of the genetic variance within that interval. What we did is we queried that interval in both genomes using a Syntenic graph, asking what regions are orthologous across the chromosome. And we then extracted the genes in there, aligned them, and found this particular gene, which is ABO3, abscisic acid overly sensitive 3, which responds to drought and in Arabidopsis clearly regulates the expression of many downstream genes, a really good candidate. Now, in this case, this is really simple. These are two inbred haploid assemblies that are totally collinear. So it was very easy to extract the genes of interest in this particular interval. However, if we want to transition this information to switchgrass or genetic models of grasses like Brachypodium or rice, we need a much more robust representation of synteny and a way to track it across all of these. This is when we started developing our GeneSpace software. GeneSpace builds Syntenic graphs like this one in grasses and lets you track the positions and presence-absence variation of orthologous genes across multiple reference genomes. And this essentially does what I described in Panicum hallii, but at a much larger scale, and allows you to query any number of regions simultaneously. And this is particularly important when you have genome assemblies and annotations for each of the parents in your genetic mapping population. So I'll talk about how we can use this in two examples, first in Pecan and then second across the main Maize NAM parents. So our work in Pecan started a few years back where we generated reference genomes for 4 genotypes. Pecan, like many trees, is very outbred when pollinated and is clearly heterozygous across the genome, at least in the wild. In the breeding pedigrees, sometimes there are regions of homozygosity because of small effective population sizes and inbreeding. But we built reference genomes, outbred reference genomes for each of these 4 genotypes. That means that we have 8 total haplotypes that we can compare and analyze. And this let us more effectively explore candidate genes for QTL regions. For example, there's a genetic mapping population for Lakota. Lakota is a cultivar grown in the south of the US and is derived from an F1 hybrid between two parents, Mahan and Major, which will be coded as blue and yellow for this talk. And in Pecan, like many long-lived perennial species, a lot of the genetic mapping is done in the F1 population. And this is because it takes 20 years to be able to get seeds, so you better be able to do it in 1 or 2 generations. Rills and advanced intercrosses are just completely off the table. F1 populations segregate the heterozygous variation in the parents. And so in this case, we can phase this Lakota by Oaxaca population by the grandparental alleles, Mahan and Major. And we can generate a haplotype map like this. We were particularly interested in biotic stress tolerance and gall production by various insects. You can see on the left an image of a leaf that has a lot of galls on it. And we surveyed this Lakota F1 population for gall incident. And you can see on the right of this plot, ordered by highest infection at the top to least infection on the bottom, that pretty much all of the individuals that have any gall infection at all on their leaves have this yellow genotype, this Mahan genotype on the left arm of chromosome 16. This indicates that there's a strong association between genetics and molecular variation at a region and quantitative genetic value of gall resistance. And indeed, if we apply a statistic to this and do a QTL mapping, there's a 1.2 megabase interval that likely contains a causal variant that drives gall susceptibility. So since we have an outbred assembly for Lakota, meaning that we have both haplotypes represented, we can phase our contigs in the assembly by whether they are susceptible to gall, which is the yellow haplotype, or are resistant to it, which is the blue. So we did this for this 1.2 megabase region, here depicted on the top. The primary haplotype, which is the main assembly, that's what you would normally only see in an inbred assembly, runs along the bottom, and you can see it's primarily the susceptible haplotype. There's a large gap in here, you can see right here. This is because there's runs of homozygosity in Lakota because it's a breeding pedigree. We couldn't assemble two haplotypes here because it was too homozygous, and this is done with a CLR and non-Hi-C integrated assembly. Nowadays, with Hi-Fi and Hi-C integrated, we would have a contiguous haplotype across this whole region. Either way, the take home here is that there's a number of places in this region where we have good and highly similar orthologous genes between the two haplotypes indicated by thick bars. There's some places where there are thin bars that indicates peptide differences between the two haplotypes. And then there are a lot of places where there are presence-absence variants indicated by these hollow circles and no line connecting them. The most important one is right in here. There are 15 sets of leucine-rich repeat and disease-annotated genes in this QTL interval, right in the middle of it, that are only found in the alternative haplotype. This means that if we had only used the primary reference assembly, we would have had no idea that these genes were there. And we only were able to find out that these very, very strong candidates exist in this heterozygous individual by producing and comparing an outbred assembly. This is a pretty cool way to use multiple reference genomes, but this is just two. And again, it's pretty straightforward because we can just align the haplotypes to each other and look for a specific region. But it gets much more complicated. For example, earlier this year, the genomes were published for the 26 US Maize NAM founders. Here they are presented in a gene space riparian plot where the Syntenic regions are connected across all of the genomes. You'll see that for the most part, these things are completely Syntenic, except that there are two largely segregating inversions, one on chromosome 2, you can see here, which is present in about a third of the genotypes, and one on chromosome here that's found in 2 individuals. There's also 2 private structural variants. One is this inversion for MO18 on chromosome 3, and the other is this inversion translocation between chromosome 9 and 10 for OH7B. The really nice thing about these 26 Maize NAM founders is that there are a ton of quantitative genetics resources related to them, in particular QTL for a number of traits and a number of conditions. A particularly nice experiment came from Lee et al. A few years back, where they surveyed well-watered and drought conditions for a number of traits and found hundreds of QTL. I queried their database and pulled out the 2 QTL where the effect of a single genotype was vastly different from all of the others. These outliers mean that there's some genetic variant in the focal genome that makes it different from all the others. And that we could let that be a driver of our comparison and say, okay, what are the genes that are different between this genotype and all the others? Those are the candidates. So the 2 that I pulled that were the two strongest is a chromosome 3 QTL for silking and thesis interval, where MO18 is divergent, and the other is for height, and that's the one where the Ki11 genotype is divergent. For Ki11 on chromosome 6, we can see that there are 32 what we're calling pan-genome entries. These are orthologous gene networks that are syntenic in this region. You can see that there's a number of places where there's no gene present in B73, but there is in the Ki11 allele. This means that those genes offer additional candidates for potential putative effects in this region. We'd have to do a lot more work to nail these down, but this at least expands the scope of what we know about in this region. More interestingly, on that chromosome 3 QTL, where MO18W is diverged from everything else, that QTL exists right in the middle of that private inversion that I mentioned previously. And it's interesting to note that if you look at the raw data from Lee et al., there's another ghost QTL right next to this one, indicating that maybe they didn't have a great idea of the physical position of this QTL, likely because it's driven by all of the loci that are in LD in that inversion and not by a single locus there. In fact, we didn't even know about this inversion until I did this analysis, let alone its co-localization with this QTL. So exploring these multiple genomes with a gene space approach in comparative genomics integrated with quantitative genomics has proven already to be a pretty interesting line of inquiry. So I'll leave just with a couple suggestions in future directions. First of all, I want to note that it's now possible with Hi-Fi and Hi-C integrated assemblies to build outbred genomes more effectively than inbred genomes. In fact, we would much prefer to assemble highly heterozygous genomes than partially heterozygous genomes. So go ahead and sequence your complex genotypes. They don't have to be inbred. Also, it's really important to work hard on the annotations. I know a lot of people know about this, but annotations are not trivial to do. You can't just lift over one annotation to another. And in fact, if you want to find new candidate genes, you really have to take this seriously and potentially use a pan-annotation framework where you use gene models from one relative to populate the models in another. Producing multiple reference genomes, particularly for parents of mapping populations, will be increasingly important. And especially in Maize and other polyploids, synteny is also an important constraint, and it lets you focus on orthologous regions and not get distracted by paralogous ones. So that I'd like to thank the organizers for having me today, our funding sources, collaborators. And I'll end by sending out a shout out here to our CROPS conference. This is held in Huntsville, Alabama at Hudson Alpha, where we focus on using genomics to advance crops and crop breeding initiatives. This is a great conference, 150 to 250 people. It's really fun. I hope you'll join us. Thank you.